Hi guys, so here's your How to Survive for Leah Smith. So first thing you'll notice is that while the page numbers might seem high, the amount of material, and I mean by discipline, is a lot lower. So it's really just immunology, microbiology, and pharmacology. So less breadth. We are going to cover in immunology, we're going to cover effector functions of B cells, and namely the different classes of antibodies, which some of you may be familiar with. If not, we'll go through some salient points there. In micro, we're going to start talking about some of the weird bugs. So a lot of times I've talked about like gram-positive or gram-negative, and the world just isn't that simple. There are plenty of bacterial organisms that don't fit within this gram-positive, gram-negative structure that we talk about. And so there are spirochetes. Spirochetes are technically gram-negative, but they're so thin that you can't really see them on a gram stain. So instead you use this process known as dark field microscopy. I talk about it a little bit in the Borrelia Burgdorferi video. While it is something that you can do, it's not a common way that we look to diagnose most spirochetal or spirochete infections. So it's not like a major take-home point, but just to give you context as to why we can't just call them gram-positive or gram-negative. A similar thing happens with a class of bacteria that we call the atypical bacteria. These, again, don't fit within that typical gram-positive, gram-negative bacterial structure. There are some organisms that are atypical bacteria that don't even have a cell wall, like Mycoplasma pneumoniae. We're not going to talk about that in this packet. We'll talk about it in a future one. But there are some bacteria that just don't fit that structure. And there are some bacteria that replicate in such a way that it's really strange. So they get called atypical, or the weird bugs. So in this packet, we're going to talk about Rickettsia, Ehrlichia, and Anaplasma, which are all atypical bacteria. And then I've got just a standard gram-negative organism and a parasite to add to your micro index. And then in pharmacology, Dr. Bradrick is going to teach you pharmacokinetics in this packet. So get ready to do some math. Okay. So introduction to Leah Smith, you have the entire case file from the get-go. You could look at it right now, and just by looking at it, if you go through the packet once, you're going to figure out what Leah has. But you also have the video here of a sweet little Leah Smith who's not looking so great. Okay, so immunology. So at this point, we have done the innate immune response, we've primed the adaptive immune response, and then we kind of took like a detour to make the adaptive immune cells, T cells and B cells. Now that we've made them and they're functional, we need to put them to work. So we're going to start from that priming stage, moving into how the adaptive immune response actually helps us. So we're going to start with B cells. One caveat to that, a lot of B cell activity is a result of T cell help. So we are going to talk about B cells first, and then if some of the story looks kind of like it's missing, just hang on tight. It'll make more sense after your KD-Diaz case when we go into T cells, and then you'll have kind of the whole picture. Something to note is that actually you're almost done with the immune response. So by the time we get to Camera Thompson Part 2, you'll be able to draw out an entire immune response. And I think by then, the whole thing will make a lot more sense. So it's okay if right now you feel like there are some missing pieces. We're going to fill them in, okay? All right. So this is kind of some background on how the adaptive immune response functions. And then we get into B cell differentiation and antibody synthesis. So remember when we formed our naive B cell and it was like ready to go out into the world, it was expressing IgM, which is the B cell receptor, and it was probably also expressing IgD so that it could leave the bone marrow. Then once it gets stimulated and is exposed to help from T cells, it becomes an activated B cell. It can then expand and it can become maybe an antibody secreting cell like a plasma cell, or an antibody secreting cell that secretes a different class of antibody. And the type of antibody and how good that antibody is at eradicating the pathogen will get better over time. And then some of them will become memory B cells, which basically just means that they hang out in the lymph node kind of waiting to see it again. They don't differentiate into a plasma cell. So I go through all of that. I go through what I just said kind of in detail here. Then we talk about the architecture of the lymph node a bit. You should have gotten some of this in your lab, but we talk about germinal centers, secondary and primary follicles. I'm not too concerned that you know the primary versus secondary follicle. It's really just to orient you to the lymph node. Things I do care about is that you know how they get in and out, so those high endothelial venules show up again, and that they're actually kind of interacting with T cells in there. So that's why the architecture of it is important, because you see that as the B cell gets closer to maturing and activating, it actually moves even closer to the T cell zone. So there's kind of, it's like an 8th grade dance. The T cells hang out in one part of the lymph node and the B cells hang out in another part of the lymph node. And as they get braver and more, interested in each other, they start to come together. Okay, so B cell activation. It requires two signals, and this is going to be the same for T cells. We don't just activate things willy-nilly. We need to really control that. So there's always a 2-step process for activation in the immune system. So here's one example, and then here's another. So I'll let you read through that, but that's important. Then we go into the different immunoglobulin classes. These are basically just different antibody types or isotypes. So this is a part of the packet you should spend a lot of time on. You really need to know the 5 different types of antibody, their main functions, and their valences. I'm not worried about subclasses. I don't really pay attention to subclasses. I actually forget how many subclasses there are from year to year, so don't bother learning that. ADCC is something that we have not covered yet, but I do kind of introduce it to the level that you would need to know for now. We're going to talk about it a lot in KDDS. ADCC means antibody-dependent cell-mediated cytotoxicity, and it's a process that NK cells use to kill virally infected cells or tumor cells. So just so that you're aware, but IgG is actually the only antibody that really works with ADCC. So you have to go through this for IgG, IgM, IgA, and for IgA, make sure you understand it has this strange structure with the J chain and the secretory component. IgD and IgE. Really the only thing you need to know about IgE for now is that it has a big job with allergens and parasites. That's as far as I would go with that. Okay, this table is a bit overly detailed, so let's just do some take-home points in the video while we're talking about this stuff. So certain antibodies are associated with certain types of T cells, and that's these T helper subsets. This is another thing that will make a lot more sense after KDDS. But really, the cytokines secreted by the type of T cell you get tells you what type of antibody you're going to get. So if you have IL-4, IL-5, and even though it's not listed here, IL-10, that's considered what's called a Th2 response, which is highly involved in allergy. So it's going to induce IgE massively. You might get a little IgG1. Interferon gamma is super pro-inflammatory. So it's going to induce IgGs and inhibit IgE. So this is like when you're sick with the flu or a bacterial infection, you really want some IgG. Th17 we haven't talked about, but I'm just showing that this is a mucosal cell type, and you're seeing a lot of IgA, which is in the mucosa. So just kind of keeping those broad points down, that cytokines actually tell the B cell what type of antibody it should make. And those cytokines are produced by T helper cells, and oftentimes they're produced by this specialty population, T follicular helper cells. So you can read about that. This is a video I made on somatic hypermutation and affinity maturation. Watch it. Understand what this is, be able to define those terms. Also, hyper-IgM is a really great way at getting at how T cells and B cells interact with each other. So I'm just going to hang out on this screen for a moment. Maybe you just want to read this. Okay. T-independent antigens. What does that mean? Basically, B cells can respond to almost anything, right? They can respond to lipids, to carbohydrates, to proteins, but T cells, they can only respond to proteins. So T-independent antigens are antigens that T cells can't respond to, but B cells can. That's kind of the short end. Read this to understand the long end. Okay, that's immuno for this one. For micro, we get the first of our vector-borne illnesses. Know this table. In fact, I think I made it one of your study questions. Just fill in this table. Be able to fill it in. Know the organism, the syndrome, the reservoir, the vector, and where you find it. Especially this column, the vectors. You have to know your vectors. Okay, enough on that. Spirochetes, I was talking about these earlier. We're only going to talk about 2. There's actually a third one, the causative agent of syphilis, which is known as treponema, but we're only going to talk about Borrelia and Leptospira today. So here's Borrelia. I'll let you go through this. Borrelia is a pretty important one. You will be expected to recognize some portions of it, probably on your readiness assessment, but we're also going to go kind of in depth on it during the case. And then Leptospira is not a vector-borne spirochete, but it is a spirochete. You actually get this from contact with animal urine, so that's a pleasant thought. So go through that and understand just kind of the background disease. Then we go into the atypicals. I would say that knowing these 3 organisms, Rickettsia, Ehrlichia, and Anaplasma, would be a good idea, particularly Rickettsia. The Rickettsia is actually an important group. Rickettsia is the causative agent of Rocky Mountain spotted fever, and it's actually the most common rickettsial infection within the United States. And the important thing to note, even though it's called Rocky Mountain spotted fever, you don't find it in the Rocky Mountains. You actually find it on the East Coast, so bad naming. Anyway, typhus. Just keep this separate. This is caused by R. prowazekii, not Salmonella typhoid. So this is a different disease than what we covered in Jenna Wilson. Okay, Ehrlichia and Anaplasma, kind of a similar tack. Just make sure you know your vectors, how you get them, where you get them. Then we go into a gram-negative organism. You can read through this. Again, know where you find it, the types of disease and syndrome. Then we have Babesia. So this is a parasite. So remember, you want to know your infectious stage, and you want to know your diagnostic stage, and you want to know your syndrome. That's the main thing. In treatment, there are some antiparasitics listed. Dr. Bradrick hasn't covered that yet, so don't worry about mechanism. I'm not going to ask you about this. This is really just FYI for later, so that if later you're looking for this, you know where to find it. But you don't really need to know anything about treatment moving into your readiness assessment. Okay, then we get into Dr. Bradrick's stuff. You're going to do a lot of math. There's an equation sheet here, and there are some terms and symbols and whatnot here. So you're going to want to check out both of those. Make sure you can access them. Then he made you a combination of videos. I've linked to the times here as well that you can follow along as he goes through. You're going to be doing equations for your self-study. If you do not understand this, I'm sorry, but I can be zero help to you. I do not math. I'm very bad at math. But Dr. Bradrick is good at it. So contact him. He knows this session is coming. We planned it for this session when it was just going to be me and him to do this. So make sure you go through this and reach out to him and be able to run some of those equations in the practice. Okay? That is all I got for you for this session. So good luck. Contact me if you need anything for immuno and micro. Contact Dr. Bradrick on all of this. All right.